Welcome to this lecture on the fundamentals of biomolecules. Today, we're going to explore a fascinating concept called Koch curves. We will learn how scientists used this elegant technique to uncover the hidden architecture of DNA long before modern sequencing machines even existed. Let us dive in. Now that we know what we are studying, let us start with a simple analogy. Imagine you are trying to put together a massive jigsaw puzzle that has been completely scrambled. If every single piece of the puzzle is completely unique, it will take you a very long time to find the matching pairs. However, if the puzzle consists of thousands of identical blue sky pieces, you will start finding matches almost instantly because any blue piece can connect to another. This is exactly how scientists study the complexity of DNA. By observing how fast DNA strands find their matching partners, we can reveal how repetitive the DNA sequence actually is. This jigsaw puzzle analogy brings us to the actual physical process used in the laboratory. In a typical experiment, double-stranded DNA is heated until it separates into single strands, which is a process called denaturation. Then, the temperature is slightly lowered to allow the complementary strands to find each other and pair back up. This is known as renaturation or reannealing. Just like the puzzle pieces, if the DNA has many repetitive sequences, the strands will find a match very quickly. If the DNA is highly unique, it takes much longer. The speed of this reannealing process is the key to unlocking the genome's complexity. So, how do we visualize this reannealing process? We use a graphical representation called a Koch curve. The unusual name Koch comes from multiplying the initial concentration of single-stranded DNA, denoted as C0, by the time allowed for reannealing, denoted as T. The Koch curve plots the fraction of DNA that remains single-stranded on the vertical axis against the logarithm of the Koch value on the horizontal axis. This specific plotting method creates a signature S-shaped curve. By analyzing the shape and position of this curve, we can directly measure the complexity and repetitive nature of any DNA genome. Before we look at the math, it is important to understand why this technique was so revolutionary. Before the invention of modern DNA sequencing machines, Koch curves were the absolute primary tool scientists used to understand unknown genomes. They led to the groundbreaking discovery that eukaryotic genomes, like our own human genome, are not just long strings of unique protein-coding genes. Instead, these curves revealed that a massive portion of our DNA consists of highly repetitive sequences that do not code for proteins at all. Today, while sequencing is common, the fundamental principles of DNA hybridization kinetics taught by Koch curves remain absolutely essential for designing PCR primers, microarrays, and CRISPR guide RNAs. Now that we understand the significance of Koch curves, let us look at the mathematics behind them. The process of two single strands of DNA finding each other is a classic second-order chemical reaction. Because the concentration of the left strand equals the right strand, the rate at which they combine depends on the square of the concentration of the remaining single strands. We express this physical reality with a simple differential equation. The derivative of c with respect to time equals negative k times c squared. The negative sign is crucial because it indicates that the single-stranded DNA is disappearing as it successfully forms double strands. The constant k is the second-order rate constant, which depends on environmental factors. While that differential equation captures the moment-to-moment speed, we need a different form to plot our experimental data. By integrating the differential equation from time 0 to time t, we get an equation that directly relates the fraction of remaining single-stranded DNA to the COT value. The ratio C over C naught is exactly what we plot on the vertical axis. The product, C naught times T in the denominator, is the COT value plotted on the horizontal axis. As the COT value increases over time, the denominator becomes larger and larger. This causes the fraction to approach 0, physically meaning that all the single-stranded DNA has successfully paired up into double helices. This integrated equation leads us to a very important reference point on any COT curve, known as the half-COT value. Conceptually, this specific point occurs when exactly half of the single-stranded DNA has successfully renatured into double strands. If we plug the fraction 0.5 into our integrated equation, it simplifies beautifully to this formula. The half-COT value equals 1 divided by the rate constant k. The symbol COT 1/2 represents the specific COT value at which 50% of the DNA has reannealed. The symbol k is the rate constant. Because complex genomes have a very low rate constant, they will naturally have a very high half-COT value. Therefore, this value acts as a direct mathematical measure of genomic complexity. Before we move on, let us see this formula in action with a concrete example. A researcher finds that exactly half of a newly discovered viral genome has reannealed at a COT value of 0.05 mole seconds per liter. We are asked to find the second-order rate constant. We know that the point where half the DNA has renatured corresponds perfectly to the half-COT value. We simply rearrange our formula to solve for the unknown variable k, which equals 1 divided by the half-COT value. Substituting 0.05 into the denominator gives us a final answer of 20 liters per mole per second. This relatively high rate constant physically means the viral genome is quite small and simple, allowing strands to zip together very rapidly. Now that we have calculated a rate constant, let us look at how we compare two different organisms. For genomes that consist entirely of unique, non-repetitive sequences, the genomic complexity is directly proportional to the half-COT value. This means if the genome size increases by a certain factor, the time it takes for half the DNA to renature will increase by that exact same factor. We express this with a simple ratio formula. The half-COT value of organism B divided by organism A equals the complexity of B divided by the complexity of A. The variable n represents the total number of base pairs. If the genome size n increases, the half-COT value must increase proportionally, shifting the curve to the right. Let us apply this proportional relationship to a real comparison. We're comparing a simple bacterium, organism A, with a complex yeast, organism B. We are given that the half-COT value for the bacterium is 10. We also know the yeast genome is exactly 50 times larger and more complex, meaning the ratio of their complexities is 50. We are asked to find the expected half-COT value for the yeast. We set up our ratio equation and substitute the known values. Since the right side of the equation simply becomes 50, we multiply the bacterium's half-COT value of 10 by the complexity factor of 50. The final answer is 500 mole seconds per liter. Physically, this means the yeast DNA strands are swimming in a much larger pool of unique sequences, making it 50 times harder to find their matching partner. Now that we have calculated these values mathematically, let us visualize them on a standard COT curve graph. On the vertical axis, we see the fraction of single-stranded DNA remaining, starting at 1.0 and dropping to 0. The horizontal axis shows the COT value on a logarithmic scale. Notice the dashed horizontal line at 0.5. This represents the half COT point we just calculated. You can see three distinct S-shaped curves. The leftmost curve represents a very simple, highly repetitive synthetic sequence, poly-U, poly-A, which renatures almost instantly. In the middle, we have the E. coli bacterial genome. Furthest to the right is the unique DNA from a calf thymus, which is highly complex. This diagram beautifully illustrates our key concept. As genomic complexity increases, the entire COT curve shifts further to the right. This brings us to the end of our exploration. So let us summarize the key takeaways. First, COT curves are an elegant way to measure the hidden complexity of a genome simply by watching how fast its DNA zips back together. Second, the absolute most important metric is the half COT value, which is inversely proportional to the rate constant. Remember that a small half COT value means the DNA is simple and repetitive, finding its matches quickly. Conversely, a large half COT value means the DNA is highly complex and unique, taking much longer to reanneal. You have now mastered the fundamental kinetic dance of DNA strands, a concept that remains foundational to modern molecular biology. Great job!